Hello everyone, my name is Xiaotong Liu and I'm a PhD student at University of Minnesota. The title of my talk today is Multi-compartment Models Review Echoing of Transcriptional Response During Effector-Triggered Immunity in Arabidopsis. So what is plant effector-triggered immunity? Unlike animals or humans, the plant does not have adaptive immunity. And effector-triggered immunity is one major mode of plant immunity. When the bacteria attacks the plant, it can secrete some effectors into the plant cells, and the plant is able to recognize those effectors and triggers a series of downstream response to defend itself from pathogen attack. Among different types of effectors, AVR-RBT2 is a typical effector protein. There is a massive transcriptional reprogramming observed during effector-triggered immunity in Arabidopsis. In one of the studies, Mai et al., more than 2,000 genes are identified to be significantly induced at different time points post-AVR-RBT2 treatment compared with mock, as shown by this heat map. In our study, we took the time series transcriptome data upon infection with Pseudomonas syringae DC3000 expressing the AVR-RBT2 effector from Mai et al. There are two treatments, including AVR-RBT2 and mock. 8 time points post the treatment with 3 biological replicates, and we will be able to get a transcriptome including 2,300 genes. In order to interpret the transcript dynamics, we developed these multi-compartment models. There are 2 major steps for MCM. The first step is to generate a chain of input compartments. So we generate compartment 0 with an exponential function. And the first compartment is linearly activated by the input 0 and is associated with a first-order decay. Based on the same idea, we can extend this input chain up to 6 compartments. And the early compartments tend to have early peak response. Later compartments tend to have later peak response. The second step is to feed the temporal transcriptome data with a 2-input combination. So we took 2 of the input compartments as the input for the output compartment, and we assume negative binomial distribution of the transcript data, and based on the maximum likelihood estimation, we can characterize those parameters by fitting the data to that differential equation. After fitting the data, we get more than 1,800 genes that are well fit with MCM. We generated the synthetic profiles by sampling from the early fit curve and the late fit curve at 7 different time points spanning 0 to 24 hours based on the MCM fitting. And surprisingly, we found that the timing and the amplitude of the second peak response for each gene echoes the dynamics of the first peak response for the majority of the genes. We proposed that there is a limited proportion of the plant cells directly contacting with the bacteria and received the AVR-RBT2 effector, but the surrounding cells responded indirectly, and it is very likely that the first and second peaks largely represent the responses of type 1 and type 2 cell populations, respectively. I would like to thank my advisors for their help and suggestions on this work, and also we are grateful for the funding support from National Science Foundation. Thank you for your attention.